Dear children, welcome to biology online classes. So, in the previous classes, we were studying about the cell, isn't it? We have already studied about the prokaryotic cell, eukaryotic cell, isn't it? And its components we were studying. So, in prokaryotic cell in detail we have seen, and we also have studied about the plasma membrane and cell wall. Now we are going to study about the endomembrane system. So I have also already explained you that the eukaryotic cells, they are having what? Membrane bound organelles, isn't it? Membrane bound organelles are there. So that is one of the important feature of what? Eukaryotic cell that make it different from prokaryotic cell. In prokaryotic cell, how it is? There is no membrane bound organelles except what? Except ribosomes. But here in eukaryotic cells, you can find what? Different kinds of membrane bound organelles. Okay, and they all do different functions. For example, Golgi bodies, they got a different function. Endoplasmic reticulum, they got a different function. Lysosomes, they are having different functions. Okay, likewise vacuole also. But all of them, they work as, or their functions are interdependent. Okay, so together we can call it as what? Endomembrane system. Understood? So even though their functions are specific, they are having different functions. The functions are coordinated. Understood? So they are together known as what? Endomembrane system. So what are the organelles that are involved in the endomembrane systems? For example, what? Endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. And when you see the functions of mitochondria, chloroplast, peroxisomes, they are not coordinated with what? The endomembrane system. So they are not part of, they are not forming the part of endomembrane system. Okay. So let's study one by one. The first one is the endoplasmic reticulum. Okay. The ER or endoplasmic reticulum. Okay. So where you can find this you can find in the eukaryotic cells. Okay, as a network of, or network as a network or reticulum of tiny tubules, okay, that are scattered in the cytoplasm. Understood? So these what? Tube like, okay, structures which are scattered in the cytoplasm is called what? Endoplasmic reticulum or ER. Understood? And ER is having or ER divides the cellular space, intracellular space, into two different parts or compartments: the luminal side and the extraluminal side. The luminal means what? The inside ER. Okay, the site of, or the content which is present inside the ER, and then what? The extraluminar compartment. Cytoplasm is divided into, means the part which is found inside the ER is considered, or they are forming the luminal, and the extraluminal means the the cytoplasm that is found just outside the ER. Okay, the luminal or inside ER and extraluminar compartments. Likewise, it is divided. When it is attached to ribosomes, they are forming the rough endoplasmic reticulum, and if ER is not having ribosomes, then it is known as SER. Okay, so RER's main function is what? It is involved in the protein synthesis, because ribosomes are the organelles which are associated with protein synthesis. So RER what is the site of what? Protein synthesis. Understood? And you can see that they are continuous. RER mainly, you can see them, they are seen continuous with the nuclear membrane. This is the nucleus, and as the extension of nuclear membrane, you can see the RER. Understood? And smooth and endoplasmic reticulum, their main function is the synthesis of lipid. Okay, So the cholesterol or the animal cells lipid like steroidal hormones are there, and they are secreted by SER. Okay, so ER you can see as what tubules, okay, or as a network or reticulum of tiny tubules that are scattered in the cytoplasm. And what are the two different types? SER and RER. RER's main function is protein synthesis and secretion. Okay, then what is the function of SER? They are the site of lipid synthesis. Okay, and they are also involved in steroid hormones synthesis in animal cells. Okay, so about ER you understood. The next organelle is Golgi apparatus. Okay, Golgi apparatus. So Camillo Golgi has first found out this organelle, and after that only it is named this organelle is named after Camillo Golgi. Okay, so in this diagram, you can see the rough endoplasmic reticulum, which is near to the nuclear membrane. Okay, then smooth endoplasmic reticulum you can see, and you can see also the Golgi apparatus. You can see, isn't it? So, as I told, Camillo Golgi was first observed this, and they are densely stained structures, isn't it? Which are seen near to the nucleus, and they consist of flat disc-shaped sacs, or we can also call them as cisternae. Understand? And these are stacked. They means they are arranged parallel to each other. You can see many what sacs or disc-shaped sacs or cisternae are there. Understood? So the number of cisternae present in Golgi complex varies. Okay, and the Golgi cisternae, they are concentrically arranged near the nucleus with there will be a convex six cis, okay, or the forming phase, and concave or the trans or maturing phase. Okay, so the cis and trans phases of this organelle is different, but they are interconnected also. Okay, so there are concave type, okay, there are convex type what phase, that is known as the cis phase or forming phase, and there will be a concave site, okay, or concave type of surface, that is known as trans or maturing phase. Okay, and they are entirely different, but means the these two phases, they are different, but they are interconnected also. So the function of Golgi apparatus is packaging of things. Okay, the major function of what the Golgi apparatus is packaging the materials. Okay, so sometimes that the many of the molecules, they cannot be passing the cell membrane, we have learned. Okay, so this Golgi apparatus helps in the packaging of material so that it can pass the cell membrane. Understood? So materials to be packed in the form of vesicles, okay, from the ER, they fuses with the cis phase of the Golgi apparatus and move towards the maturing phase. So in this way only it helps in packing. Okay, so the materials that are coming from the ER, understood? So to that is needed to be packaged or to be sent to different parts of the cell. First, what will happen? They form vesicles from the ER and fuse with the cis phase of the Golgi apparatus and then move towards the maturing phase. Okay, so this is how I told Golgi apparatus and endoplasmic reticulum, they are part of endomembrane system. Understood? So why you understood now why the Golgi apparatus are seen close association with ER. Okay, then a number of proteins synthesized by the ribosomes on ER that are modified in the cisternae. Understood? Before they are released from its trans surface. So modification of proteins, packaging of proteins, okay, and these are the site of formation of glycoproteins and glycolipids. So these are the major functions of what Golgi apparatus. So Golgi apparatus, where you can see in the cytoplasm, okay, near to the nucleus. And they also contains what? They contain the network, isn't it? They contain networks of disc-shaped sacs or cisternae. Understood? And these cisternae, they are packed parallel to each other, and the number is of the sacs varies. Okay, and I told there are two phases: cis phase or the forming phase, and the trans phase or the maturing phase. Now I told what are the functions of Golgi apparatus: packaging. Okay, then what it helps the proteins produced by the ribosomes to be modified, okay, before they are getting delivered, released from the trans phase. Then it's also forming a part in the formation of glycoproteins and glycolipids. Clear? So next organelle is very what a very famous organelle. We have studied that in ninth class also. So they are known as the suicide bags, isn't it? Or lysosomes. Okay, so what are lysosomes? They are membrane bound vesicles. Okay, formed by process of packaging in the Golgi apparatus. Understood? So in the Golgi apparatus, the vesicle like structure get packaged. Okay, and this membrane bound vesicles or bags are known as what? Golgi apparatus. Okay, so the major thing that you are finding in a lysosome is hydrolytic enzymes, various hydrolytic enzymes like lipases, proteases, carbohydrates, etc. So what are lipases? They are the enzymes which they help in digesting lipids. Okay, and proteases are the enzymes which help in the digestion of proteins, and carbohydrates are the enzymes which are helpful in digesting the carbohydrates. Understood? So this kind of enzymes are filled in this. So and this will be active at the acidic pH, means in the low pH. Okay, so these enzymes can digest the various components of cells. So cells are made up of what? Carbohydrates, proteins, lipids, nucleic acids, etc. So this lysosomes contains what? The various hydrolytic enzymes, and with the help of this they can digest the what? Various materials. Clear? So they are known as what? Suicide bags. You know why they are known as suicide bags? Because what? These enzymes, when a cell is damaged or when a cell is affected by the any infections, okay, this lysosomes can burst and can release the enzymes of them, and this enzymes digest the cell content, okay, or the what to say, the infected cell. Is it clear? So this is the what? Another part of another endomembrane system. What is that? Lysosomes. Next is what? Vacuoles. So next organelle is vacuoles. Okay. So vacuoles are what? Membrane bound space in the cytoplasm. In the cytoplasm, there are certain spaces which are covered by plasma membrane or cell membrane. Okay. So this, what it contains? It contains water, sometimes cell sap. Okay. Then excretory products like this which are not useful for the plants. Okay, so the vacuole is bound by a single layer of membrane, and that membrane of vacuole is known as the tonoplast. So what is tonoplast? The membrane of the vacuole is called what? The tonoplast. Okay, and you know the difference between the vacuoles of plants and animals. In plants you can see a central large vacuole, isn't it? The cells 90 percentage of the volume also can be occupied by a vacuole. Understood? But in animals, how it is? They are small in size, and they'll be more in number. But in plants, what? The number is less and the size is large. Okay, and in plants, what is the function of this vacuoles? The tonoplast, what is a tonoplast? The membrane of vacuole is called what? Tonoplast. So they facilitate the transfer of ions and what various materials across the or plasma membrane against concentration gradient, understood, into the vacuoles. So against the concentration gradient, what can take place? Transport of ions, understood, and for this tonoplast is useful. Okay, so the their concentration is higher in vacuole than the cytoplasm. Okay, so even though they are what not useful, okay, they are helpful in what in maintaining the ionic balance etc of the cytoplasm, because they can be stored in the vacuole, and when it is needed they can be transported across against the concentration gradient, understood. So this is the reason why the concentration of this kind of things are things means what the content of the vacuole, okay, they are having what they are having more concentration, isn't it, than the cytoplasm. Okay, so in amoeba there are contractile vacuole which can help for osmoregulation and excretion. Okay, and there are cells with the food vacuoles. Okay, so they are formed by engulfing the food particle. When food is engulfed into the cytoplasm, they seen as what? The food vacuoles. So about vacuoles you understood now. Vacuoles are what? The membrane bound space. Okay, and in plants it is big, large, and their function is what? They store various materials. Okay, and they help in the transport of ions and other materials against the concentration gradient, means from lower concentration to higher concentration into the vacuole. Okay, so this is the reason why vacuoles are having what? More concentration. Okay, concentration will be high in the vacuole than in the cytoplasm. So this much endo membrane systems includes what all? The endoplasmic reticulum. Okay, they are of two types I said. Golgi apparatus I told. What is the speciality? They are having cis and transphase, isn't it? Then I told you about the lysosomes and vacuoles. Hope this much is very clear. We will continue with the other topic in the next class. Thank you.